Well, here we go on chapter 22. Chapter 22 is about the lymphatic system, something that anatomically caught our attention because it contains this clear liquid that we find all over the body as a special set of vessels. Unlike the circulatory system, which sends the blood out to the capillaries and then returns it, the lymphatic system collects fluid in all the tissues. In fact, the ends of the vessels are right in the middle of a capillary net, a capillary bed, collecting the excess fluid that escapes the circulatory system at every capillary bed and returning it, balancing that fluid flow. So basically, this vessel system is just going to collect and return. We're first going to go over the anatomy of the vessels, the organs, and the tissues that we identify as part of the lymphatic system. We'll talk about its importance for fluid balance, the overall avoidance of edema. You think about it, when more water leaves the capillaries than is returned, eventually that would cause the tissue to swell, and it's only the drain, the sort of sump that's provided by the lymphatic vessels that returns this to the blood system. But by far the more interesting and more recognized function of the system is that of defense of the body. We have a number of defensive systems working independently. They have overlapping effects, and they are effective against a diverse array of threats. Now, going back to the fact that it's based on protein chemistry, so our proteins are interacting with the proteins of other living organisms, the proteins of supermolecules like viruses, threats such as toxins. We can reduce the toxicity of a poison using some of our proteins in the immune system. Being able to attack organisms that are multicellular organisms. So when you're attacking an intestinal worm, or the larva of a botfly, or a subcutaneous parasite, you're attacking a much more extensive living system than a single bacteria. So let's start with the anatomy, and then we're going to break down the defense into two categories. Innate means inborn, and actually we're going to start with the integumentary system. The first job of the integumentary system is keep it out, protect the inner tissues, and that's part of our innate immunity. But there are other cellular and chemical mechanisms that we're born with, which means they're loaded and ready to go when you're born. They do not require the experience outside the mother in order to activate them. Adaptive immunity, you have the capacity to become immune, but you only become immune through the experience. The experience can be an infection, it can be a disease. It can also be a vaccination. We have found less threatening ways of activating our immune system against specific threats. The anatomy of the lymphatic system begins with its circulation and drainage for water balance. And so we see here a very kind of odd distribution. Look over here at this diagram. One set of converging lymph vessels. By the way, lymph vessel is synonymous with the term lacteal, the smallest lymph capillaries. But you'll notice that this upper right quadrant of the body, the head, neck, and thorax, and the shoulder, arm, hand, and wrist, is drained by one system. It basically returns that fluid to the right lymphatic duct, here by the right subclavian vein. The rest of the body, 3/4 of the body, is a system of vessels that converge on this thoracic duct, the main duct here in the abdomen, spanning up into the thorax, where it basically deposits its lymph back into the returning blood on the left subclavian vein. So it's a kind of odd 1/4, 3/4 distribution. Whereas the circulatory system has diverging vessels to capillary beds and then converging vessels because the back to the heart, because the heart's propelling the blood out and back. The lymph vessel is going to start right here in the tissue and collect the extra fluid. That fluid is going to be moved by milking action, the random squeezing that comes from the motions of the body, and one-way valves are going to direct that flow one way back toward the return points here high in the thorax. So the typical view we would have of a capillary bed now has to include this green system of the ends of these lymphatic capillaries. You can see that the fluid comes from the tissue itself and is collected in these capillaries, and milking action moves them out. This water movement to drain the excess fluid and maintain water balance is a primary job of the lymphatic system. It's performed so well that it often is not discussed. So when we see these tubes transiting a certain area, it makes sense that they are often bound with the arteries and veins of the region. So you see here the profile. Here is the artery, more circular and small and thick-walled. Here's the vein, thinner-walled and larger, often with an irregular shape. Here's the lymphatic vessel, very thin-walled, with gaps between the cells, which I'll show in a second. And this irregular cross-section, its large diameter then means that it can hold quite a bit of fluid and conduct it away with little resistance. Here's what a lymphatic capillary or a lacteal will look like. To me, this always, I'm a botanist, so it makes me think of bamboo, where we have segments that are linked together to produce the tube. But a couple of features of the lacteal deserve mentioning. First of all, these thin epithelial cells overlap one another and overlap one another tightly enough that liquid like lymph, and lymph is a clear liquid, can actually flow between these cells through the gaps and accumulate here in the lacteal. Now, the serotic tissue is moving and will squeeze down on these plates, causing pressure and causing the fluid to move. Now, the fluid cannot move in the opposite direction because back here where the pointer is, there's a one-way valve, and that pressure is pushing against the backside of the valve. But here, there's a ready opening. This lymph flows forward until the pressure is released, and then the backflow closes this valve. So basically, the lymph is moving kind of in a stepwise motion. Squeeze this chamber, and you move the lymph to the next chamber. Squeeze that, and you move to the next one. Wherever lymph is accumulating, the lymph vessel gets larger, and consequently, smaller pressures will move the lymph along. Here's an actual micrograph showing you this. This kind of reminds us, I think, of veins. But back here, which is the origin of the lacteal, there is not a continuous path in that direction back to some pumping organ. There is an end. The lymph flows along here, and you'll notice this characteristic of a one-way valve. The one-way valve allows flow left to right. It will prevent flow right to left, but as it does that, the pressure kind of causes this bulging around the valve. That's the process that leads to varicose veins in older people in the cardiovascular system, and it's a characteristic of the lymph system as well. But because of the pressures, there's no pumping organ, and the milking action is less, actually applies less pressure. There's a little less bulging. Now, along the way, here we are making our trip back. Here's a lacteal network, 1, 2, 3, 4 places in this drawing where lymph is entering this enlargement. This is a lymph node, and lymph nodes are arranged all over our body to collect the flow from multiple smaller lacteals and to screen it. You can kind of see the idea up here. As the lymph flows in, lymph is a wonderful communication network. Our protective cells are found there all the time. But it's also a wonderful pathway for pathogens and for toxins. So when we deposit this lymph into the inner structure of the lymph node, it's going to be filtered through these different functional areas. And in these areas, we're going to find specific assemblies of different protective cells. Now notice that this lymph node is an active organ. Here's the blood flow, delivering blood to all the capillary beds, which are not drawn in, and returning the depleted blood from a vein. That is typical of something that has living cells, because we're having to supply oxygen and nutrients and withdraw waste from all of these living cells in the lymph node. You can see that these partition divided into different capsules that basically are going to divide the flow as it collects, and eventually it's going to move it out. But in here we notice a couple of repeating structures. Within this subunit, this region, these are called tuberculi, and they produce a kind of a capsule within a capsule. And here is a dotted region called outer cortex. This is full of B cells. So pathogens can stimulate B cells to activate and to produce antibodies and to release them, so that the arriving pathogen encounters a defensive fight, a defensive contest right here in the lymph node. Now, this is something that you sort of are familiar with. You know, when you get a respiratory infection, the colds or flus, and your lymph nodes swell and they become tender, especially here in the neck. That swelling is from the attempt of the body to restrict flow out of the lymph node. So this becomes pressurized, it swells up, the stretch receptors are sending pain messages, and pain is generated. But a lymph node is a place that can try to contain the arrival of a pathogen and deal with it directly before it spreads further. So you can see the multiple B cell loci that are present to combat arriving threats. This final ring, this donut-shaped dotted line, is a location called deep cortex, and the deep cortex is populated with T cells. Now, T cells mature in the thymus, and you remember they're cell-mediated immunity, so they're the cells that are armed with specific antibodies and are actively, these cells, the active cell, the living cell, called the cytotoxic T cell, must be present to provide the defensive effect. Now, the organs of the lymph system. Here on the front of the heart is an organ which is a mystery to me. Look at that organ. It's very obvious. It's large in extent. It has that kind of lobed appearance like cottage cheese of a glandular organ. But I noticed in all of our models in the lab our mini torsos and our full torsos, they don't even model the thymus and put it in place. It's right here in the central part of the mediastinum, above the pericardium and heart, between the pleural membranes and lungs, right where the major vessels, where the trachea, where the esophagus are present. A very important region for the thymus in relation to other organs of the body. Now, the thymus is interesting because it is actively growing and metabolically active when you're young. And it kind of stabilizes as you reach an adult life and becomes somewhat less active, although T cells continue as part of our defensive system. It kind of leads me to think that immunity is something that's more active when you're young. When you come out naive and not protected against anything, is when you develop your immune system. You initiate your immune responses. Now, what does that mean? That means that early conditioning may have wide-reaching effects on the vitality and the activity of your overall immune system. Like other systems, there may be a maturation or a conditioning period which is most active when you are young. One interesting feature of the COVID-19 virus, and now we're in that middle stage where some of the things that were said earlier are being contradicted. So I'm not sure about the currency of this statement, but it seemed at first that young people, healthy people, people without health issues, were experiencing no symptoms or very mild symptoms of flu-like symptoms for COVID-19, whereas people who have pre-existing problems and old people are experiencing more serious threats to their life. Kind of a curious thing, the thymus. The spleen is the other major organ that we have for the lymphatic system. Here, right at the base of the xiphoid process is where this transverse section has been cut. And it's also been spun around. The spinal side is inferior. So here are the vertebra and spinal cord. On either side, the kidneys are right at this level. And over on the right side is where we find the spleen. The spleen is within the peritoneal membrane. This is a shape that, like the other organs, here's the stomach, here's the liver, this is a space, the abdominal space, where these organs are growing together and filling the space. And so basically the shape of this spleen is managed and affected by the shape of the cavity wall, the growth of the stomach, the fat packing that you see, the presence of other membranes. Let's now talk about lymphoid tissue specifically. And the overall anatomy is very straightforward. Let's get to the function. First of all, I want to make a distinction between primary lymphoid tissue and secondary tissue. The primary lymphoid tissue is where the immune cells are produced and where they mature. And that includes two tissues, bone marrow and thymus. Now, this is curious to me. We talk about a lymphatic system and lymphoid tissue, and yet the bone marrow, the red bone marrow, part of the skeletal system, is the source of all of our white blood cells. So they originate in bone marrow. If they mature there, they are released from bone marrow as B cells, and they are in the general circulation. They may accumulate in lymph nodes. We will find them in connective tissue throughout the body. If these pre-T cells that developed again in the bone marrow move through the general circulation to the thymus and mature there. They produce T cells, and that means that bone marrow and thymus tissue are the primary tissues. The secondary tissues is where the immune cells do their work, where they actually confront and deal with the antigen. So that means lymph nodes, places like the spleen, which is a tremendous screening organ. The blood circulation and the lymph circulation through the spleen allows the collection of immune cells there to detect the presence or signs of the presence of infections and begin the immune response. Tonsils, which are in the major opening up here at the top of our body, because it's so much of an opening, and it's an opening that has to deal with entry and exit all the time. It's difficult to close it down, or we have to breathe all the time. We are eating and drinking all the time. Because of that, it's an area where the prospect of invasion and infection is very great. So tonsils are really enlarged and specialized lymph nodes. After that, I want to name two abbreviations, MALT and SALT. In some cases, the lymph tissue, the secondary lymph tissue, is not extensive enough to actually name it as an organ of the system, but it is a tissue that is existing in another named organ. So, mucus-associated lymphoid tissue. Remember, mucous membrane lined all of the cavities and pathways that open to the outside. So the straight through digestive tract, the in and out respiratory tract, the reproductive tract, which is mainly an exit tract. Those cavities that need protection, like eye and ear, will have mucus-associated lymphoid tissue that can be there for the screening of local fluids and the arrival of potential pathogens. Likewise, our outer covering, the skin-associated lymphoid tissue, is there to back up the skin's function. Its number one function, and it does it well. Keep it out. Make a barrier. Don't let it in the first place. But it is our outer surface. And it does have openings, just like the gates of a fort require special reinforcement and special protection. All of our openings must be protected. And so the skin associated with those surfaces and those openings have lymphoid tissue in order to screen for arriving threats. Now, on the cellular level, all living things are made of cells. If we have a system, an organ system, it's about what cells are and what they're doing. And we recognize now more and more specialties among these cells. What's shown here are kinds of lymphocytes, our adaptive immunity cells. The T cells, or the cell-mediated immunity cells, are about 80% of the circulating lymphocytes. This is a cell that's armed with antibodies that will basically be on patrol as a living cell to detect potential threats to the body. There are 4 types listed here, and I want to first name the cytotoxic T cell. This is the direct attack cell. If you've got something that's attacking a foreign pathogen, like a bacteria, a foreign pathogen, it can be a eukaryotic cell. There are several diseases caused by eukaryotic microorganisms. If it's a T cell that can recognize a virus-infected cell and eliminate that cell, then it's called cytotoxic cell poison T cell. This is the direct attack. Over here is the cell that's left over after the infection has been cleared. A T cell that's a memory cell. It is retaining the information to make the specific antibodies. and to be prepared to react to that specific disease. So this would be the memory cell that results from having smallpox or from being vaccinated. It will prevent, absolutely prevent, the explosion of a smallpox pathogen infection, and basically eliminate it when you're infected prior to you noticing those symptoms. So in a way, we say, well, I don't get smallpox anymore. Well, actually, you do get infected. But the memory cells react so quickly that it's eliminated before the cell population gets high enough to be a problem. Now, I mentioned smallpox. There's a little bit of a difficulty with mentioning that because we have declared smallpox an extinct disease. We're now having an argument about should we retain our cultures of smallpox cells? Can we eliminate all of them? Since it's gone, is it safe to stop inoculating people for smallpox? Well, I very seriously doubt whether the human carrier is the only repository for smallpox. And just 2 years ago, there was an interesting report. People were cleaning out a number of old labs in a medical school. Some labs had fallen into disuse and had accumulated various collections, boxes, and so forth. And in going through some of those boxes, they discovered tubes that were labeled as microorganisms. And among those tubes were 6 tubes that had the smallpox pathogen. When they were examined, it was discovered that 2 of them still had cells that could be revived. That kind of example kind of makes me cautious about this disease, which was so damaging and lethal to human beings for such a long time. Now, here in the middle, these are not direct attack cells, but they are cells that directly affect the homeostasis of T cell action. Helper Ts are sort of like scouts. They are sort of like, I don't know what, T cell counselors. In the presence of helper Ts, the activity and the sensitivity of cytotoxic Ts go up, and it bridges over to B cells to provide other sources of immunity. Helper Ts magnify the effects of the immune system across the board. In fact, helper Ts, this is the cell that is adversely affected by HIV infection. What we call AIDS, the disease AIDS comes when HIV has attacked and eliminated the helper Ts. So you don't die of AIDS. You die of something that normally, if you're diagnosed with pneumonia and you have a healthy immune system, you may need to go to the hospital, but you typically can recover from it with medical treatment. But without these helper Ts, the pneumonia is going to kill you. Now, if you can enhance the sensitivity, you need to also be able to reduce or suppress the sensitivity. And that's what these suppressor Ts do. This is something we've seen in homeostasis. We typically have complementary mechanisms where we can raise a concentration if it gets too low, or lower it if it gets too high. And that's what these 2 address. Now, I would mention that in addition to confronting real pathogens that cause real disease, we do have the instance where our own immune system becomes hypersensitive. So oversensitivity to the environment is what produces allergies. It's what produces anaphylactic shock. And it's what produces chronic respiratory diseases and so forth. So we do have to manage our respiratory system within a range of effectiveness. Over on the other side, The other classes shown here, we kind of characterize B cells, those that mature in the bone, as those that produce and release antibodies. And this is showing the circulating B cell when it's not stimulated, when it's not activated. But when it's activated, they differentiate into this much larger cell called a plasma cell that is just full of endoplasmic reticulum and Golgi apparatus. Those are the synthesis organs of the cytosol, the cytoplasm, and these are then constantly making the antibody and releasing it into the fluids of the body. This cell doesn't have to be there to attack and neutralize the antigen. The antibody does it independently of the cell. Now, natural killer cells are shown here, over here, because for various reasons they think they're more similar to B cells, but they are distinct. This is innate. We're born with natural killers that are ready to go. Kind of your genetics of your fertilized egg is determining how your natural killers will behave. They're about 10% of the lymphocytes. And one of the things that's fascinating about them is the ability to recognize a self cell, one that's infected with a bacteria or infected with a virus or in some way compromised, and attack it and eliminate it. Back to the red bone marrow, here we are, the skeletal system being the source of all our white blood cells. The differentiation in the cells have their own developmental pathway and their own distinguishing factors. So a hemopoietic stem cell is multipotent, and a particular colony stimulating factor is going to basically determine what kind of stem cell it forms. The lymphoid stem cells are those that lead to natural killers and B cells, which is why we kind of think of this pathway when we think of natural killers. The thymus is where we get thymic hormones as the differentiating chemical factors that cause these lymphoid stem cells to produce T cells of all types. Now, this is, as we said, the cell-mediated immunity. So we talked a lot about the cells, the tissues, the organs, the anatomy. We've also mentioned again and again that what we're basically talking about is a defense. All the weapons are made of proteins. That means that genes in our nucleus code for those weapons, those that we receive from our parents at conception. All three types circulate through the bloodstream, establishing the immunities that we were born with or that we naturally acquire. In addition to natural acquisition, we can acquire immunity in artificial ways by vaccination. We'll look at those different mechanisms a little bit later. So first, if it's a natural killer system, we call it immune surveillance. And the idea is that the natural killer cell, with its recognition proteins on its membrane, react to an abnormal cell. In addition, in this case, these red dots tell you something is in that cell or going on in that cell. Maybe this is a cancer cell. Maybe it's infected by a virus or a bacteria. In a sense, we call these natural killers because they recognize, attach, and destroy this abnormal cell. This is a remarkable discovery. This is self-cell monitoring. This is body cell elimination. In fact, for those lethal cancers, this has been the big problem. How do you selectively kill a cancer cell, which is one of your cells, when it's nested in among a group of cells, all of which are genetically identical? one of the hardest medical tasks we confront today. But it kind of shows you the approach we're beginning to take, and also should give you a clue to the important diagnostic step when we do cancer. So, for example, breast cancer is a leading cause of death among women, and in the 1950s was a virtual death sentence. However, now, upon the detection of a cancerous growth, they do a diagnosis that characterizes the genetic nature of the tumor, because there are several tumor types that we have developed a specific or immune-based chemotherapy. These are less dramatic in terms of their side effects and more effective, so that the chance of recovery is much greater if you have an immune therapy developed for your type of cancer. If you have what's called a triple negative, then the general cancer treatment, chemo, is based on the fact that cancer cells divide so quickly. And so you're going to basically treat the entire body, hoping that it reduces and eliminates the cancerous tumor or any remnants of it. Now, during that process, this general effect is going to affect all parts of your body that are rapidly dividing. So hair production is rapid, nail production is rapid, skin is affected. Because the digestive tract is constantly suffering abrasion from moving solids through it, its lining is actively dividing, and so you get nausea reactions, you get diarrhea and other unpleasant effects. And the difference, and I think the hope for the future, is the ability to diagnose more closely what these different cancer types. And the breast cancer is only a cancer that occurs in the breast, and there are different genetic types. So as we develop more and more specific immune therapies, cancer treatment will be more successful and actually much, much less unpleasant to endure. Over here, antibody-mediated immunity. This is the B cell and plasma cell, and here are those antibodies. Notice again, they're Y-shaped. A basic stem, which is sort of the superstructure that you hang it on, and then two arms, two binding sites that allow the antibodies to bind to antigens. Over here is the T cell sequence, with the cytotoxic T being the active agent against, in this case, an abnormal cell. Back to this idea. A cell is produced. Now, in the bone marrow, this hemocytoblast is going to basically produce a blood cell, the beginning step of a blood cell. After you differentiate from the true stem cell, the daughter cell, to this, you're going to become a blood cell under the influences of these colony-stimulating factors. And we've already been through this. One line to the lymphoid stem cell will lead to lymphocytes. Another line to the myeloid stem cell, the different cell factors like EMO, like GM, will differentiate. We have no colony-stimulating factor identified for the platelet line. If it's not in the presence of EPO, shun it off to a red blood cell, or the presence of this GM CSF, differentiated between general and monoblast cells. These are the white blood cells that are important in the lymphatic system and our defense against infection. All right, time to break this down. Innate means you're born with it. It's genetically influenced by the genes you got from your parent. That's your shot at innate immunity. It will then work on those genes based on your whole life. If you lack genes that other people have, you will lack some of these properties. It works basically on pattern recognition, which is based on the genetic identity of all your cells. Every one of these cells is going to have exactly the same nucleus, exactly the same genes as the fertilized egg, and exactly the same as the white blood cell that's producing the immune reaction. The other category is specific acquired immunity. Specific acquired immunity is built up through your experience and the way your genes react to it. So you get the capacity at fertilization, but the actual immunities that you develop depend on your life, your region. So we live in northern temperate regions, and we experience a certain number of diseases routinely. Those diseases like malaria or cholera or impetigo, basically these are diseases that have been controlled by the combination of our civil engineering and our medical discoveries. But those things that you experience will develop your adaptive immunity. Let's take a look at the breakdown. First of all, here's innate. And there's basically two very different strategies represented on this list. This top line, physical barriers. This is your skin. This is the protection of your openings. And the idea is just deny access. Keep it out. Physical barriers are the coverings. They are the linings of your openings. And they are a feature of innate immunity. All of the others, from here down to here, are cellular based. And fever is actually a reaction that's based on a cellular product. So let's just name them. Phagocytes, our white blood cells, can eat invading pathogens. Immunological surveillance, what we talked about with our natural killer cells, recognizing invaders. Interferons, a new concept, and the idea is this. Interferons can prepare uninfected cells to react to infection. So they're produced by an infected cell, and interferons will arm uninfected cells. If they are not infected, they continue to live along. But if they are infected, that cell bursts. It self-destroys, limiting the spread of the disease. An interferon effect. Complements are secondary genes that produce proteins. These proteins act in a complex way to identify pathogens. In some cases, they might label a bacteria with complement, and when labeled with complement, now all of a sudden they're much more apparent to the phagocytes. So there may be interaction between these lines. Inflammatory response are the multiple cellular effects that come when mast cells and others release the damaged chemicals like histamine, heparin, prostaglandin. And they basically activate many features of the body that contribute to limiting the spread and healing of the injury. Finally, fever, the resetting of the body's thermostat, takes advantage of the fact that free-living microorganisms may have to tolerate a really wide range of temperature. You think of bacteria that's living in the soil. If it's living in the upper layers of the soil, it's going to have to tolerate our 105 degree Fahrenheit summer and our 10 below Fahrenheit winter. But when a bacteria can live in our body, it basically specializes on our body temperature. It grows and reproduces best at 98.6. So it turns out that if you raise the body temperature just a little, a couple of degrees, by 100 degrees, the division rate of those bacteria in our body goes way down. And what it basically is trying to do is slow those down so that our body's immune system has time to operate. The non-self recognition that goes on in all of these systems below the skin is very simple. If it's not me, I'm going to kill it. I'm going to grab it. I'm going to take it out of circulation. So that pattern recognition is important to all of these built-in and ready-to-go systems. First line of defense, keep it out. This won't take long because physical barriers provided by the epidermis, made of epithelial tissue, by the understructures of the dermis and hypodermis, make the skin itself dry on the surface, covered with salt, covered with oil, inhospitable under ancient conditions to bacterial growth, and basically impermeable. Around our openings and down our pathways, like this might be the urogenital tract, this is the respiratory tract, here is the adjoined digestive tract, and other vulnerable openings that are moist and subject to chemical invasion, the eye. Those are openings and surfaces that must be protected, and that's where chemical effects are going to have an antimicrobial effect in general, but hopefully also an antipathogenic effect, so we don't get those organisms that don't treat us well. Now, let me just, before I go on, let me make a point. The large majority of microorganisms do not live and grow well in our body. There is a large group of microorganisms that can grow in our body without apparent negative effects. So the gut flora, as it has been called, are microorganisms that routinely live in our large intestine. We used to say there are tens or dozens of species in a healthy gut flora. We now know from DNA sequencing that it's much greater. It's hundreds of species or more. In fact, in some cases, a particular bacteria is an element of our homeostasis and of our health. So it is just those that produce a negative effect that we call pathogens. So back to the skin. Here's our revisiting our integumentary system with this stratum corneum, these flattened, dead, keratinized cells, multiple layers to aid in the absolute closure and the impenetrability of this outer surface. That's because the cells are shellacked with keratin, and they are coated with things like perspiration, which leaves salt. Salt is a great preservative. Before refrigeration, how did we preserve food? Salt was one way. We still use salt today. And sugar is another way, which basically has an osmotic effect. The cells of this continuously grow. The timing from the base of the stratum basale to the surface varies from 15 to 30 days, meaning that even in our thickest skin, we turn our skin over, completely replace it 12 times a year. We have the presence of mucous membranes in our lining, which basically makes this a lubricated and mobile surface that mucus can be moved by cilia. So we are constantly trapping particles. We are constantly trapping pathogens and moving them along for elimination and basically that holds down the infection rate. The trachea and its mucus escalator, things like peristalsis that moves things along our digestive tract and urinary tract, the processes of elimination, urination, and excretion that basically can basically push things out of the body, all contribute to keeping our tracts healthy. A couple of ideas that contribute for physical barriers is the antimicrobial action of some of our chemicals and this idea from ecology of competitive exclusion. For physical barriers, lysozyme is a substance in our lysosome, a vesicle in our cells. These are lytic enzymes that can break apart cell walls of bacteria. When we incorporate a bacterium by endocytosis, phagocytosis of a white blood cell. It is a lysosome and lysozyme release that kills that cell and begins to take it apart. The stomach is a particular cleansing organ because of its low pH, pH 2 to pH 1. And that acid is not there to decompose the food, to dissolve it or melt it away. That acid is there to sterilize the food if it's carrying pathogens down. And by the way, even with most careful selection of food sources, selection of food items, and treatment with things like cooking, we still get a certain load of pathogens arriving in the stomach. The stomach acid takes care of most of them. This idea, competitive exclusion, is based on the idea that another species grows better. So the bacterium that is present in your lower gut, your large intestine, has if it's a benign bacteria, then it's a bacteria that will grow better than mutants that might have a pathogenic effect. So the mutant or the wild type doesn't grow well in your body. The mutant or the pathogenic type doesn't grow well if the wild type is there. Now, that means that a healthy and diverse gut flora might contribute to your health. And this is really the understanding we have to predict the problem of disease resistance and antibiotic resistance. What happens when you put antibiotic down there and you have a mutant cell that also has antibiotic resistance? Now the only cell left is the pathogenic cell. And so you're actually replacing those wild types with a pathogenic bacteria and will experience more severe disease symptoms. White blood cells work by phagocytosis, a type of endocytosis that is seen especially in macrophages and neutrophils, our top killers. Macrophages are in the bloodstream or the tissue that can clean up, recognize, aggregate, clean up infection. They live for months, and they can be activated by a number of other elements of the immune system. So the reaction of B cells, of T cells, of other elements, complement fixation. All of those can make the macrophages more numerous and more active. Neutrophils are our most abundant, first on the site. They live only for days, but they basically eat until they die. They have constant killing power, and it's based on this recognition. If it's not me, I'm going to eat it. And very effective. Immune surveillance, that second idea, is based on natural killer cells. And let's just talk a little bit more about how the proteins are involved here. They will directly destroy self cells. Now that is an exception to that pattern recognition. Most of our innate immunity says, if it's one of my cells, I'm going to leave it alone. I'm not going to attack it. But natural killers have the ability to recognize an infected cell. Here's the abnormal cell shown by those red dots. We see the natural killer attach and align its Golgi apparatus, and vesicles begin to appear. Those vesicles are delivering a secretion to the junction of binding. Now down here you see what they contain. They contain a molecule called perforin, and it's the same root as perforate. These protein molecules can insert themselves in the membrane of the cell, and they form this little circle with a hole in the middle. Here you can see the insertion and the building of that circle. Once you produce that hole, the cytoplasm can leak out and this cell dies. So this is the lysis specifically of an abnormal cell. If these If these particular recognition proteins are not on this cell, then it's a normal cell, and the natural killer leaves it alone. Interferons were all in the news several decades ago, and especially because they combat viruses. They do it in a very odd way. They do it by acting on our cells. And in order to understand how they work, you have to understand what a virus does. A virus is a molecule. It's a nucleic acid. There are DNA and RNA viruses. And they're going to infect a host cell. They're going to use that host cell to reproduce the parts of the virus and assemble them. And once that's done, they will break the cell and release multiple copies of new competent virus particles. So a single virus particle infection might lead to a release of anywhere from 15 to 50 new virus particles when we break the membrane of the cell. What an RNA does is it has what's called a negative strand. It makes a complementary RNA strand that's positive, complementary base pairing. And that means we get double-stranded RNA in the cell. This RNA signals a viral infection, and it stimulates our cells to transcribe and translate the interferon genes. So we're making this protein inside the infected cell. When that happens, this is the infected cell. Here's the virus. Here's the single-stranded and double-stranded RNA stimulating the nucleus to make interferon. Well, now this cell is already toast. When this cell completes the life cycle, it's going to release more virus particles which can infect surrounding cells. But when interferon is produced, a curious thing happens. Interferon comes out to this uninfected cell and stimulates this cell to produce IAVP. Now, IAVP in this uninfected cell is inactive. But the interferon from the original infected cell is moving out by diffusion and basically treating all the cells that surround this that are uninfected. So what happens? Here we're showing an infection that's releasing 7 viral particles. So it's potentially going to infect 7 other cells. Maybe this interferon has treated some hundreds of cells in the vicinity. When the infection occurs, this infection activates the IAVP to form AVP, a new protein. And AVP causes this membrane to burst. This is programming this cell for death if it is infected. So over here, when it is infected, this cell bursts. Now let's go back to what I said a virus does. A virus takes over a cell and uses it to manufacture subunits, heads, tails, leg fibers, columns, replicate the RNA, and pack it inside the head, and then assemble the virus particle, the finished particle. Until that assembly occurs, all you've got is parts. And parts can't infect. You need the whole virus particle. So what we have here, if we break this soon enough, all we're doing is releasing the protein and RNA pieces of a virus. Without the assembly, that virus is now neutralized by apoptosis, programmed cell death. We're programming our own cells to burst if infected. And the virus spread is nullified. Now, interferons take different forms. I'm not going to go into this in great depth. Alpha is produced by cells that are infected by viruses. They attract natural killers as a supplement to resisting the infection. Beta is secreted by fibroblasts. There's a fibroblast in the collagen fiber that it's synthesizing. And what beta does is slows down inflammation in a damaged area as the fibroblast is active. Gamma is secreted by T cells and by natural killers, and it stimulates macrophage notice. So these interferons add to the apoptosis of the original diagram. Now I notice that we're ready to start on the complement system, but that we also are at 12:15, close to the end of class. So I think I'm going to stop this recording